part 2. Let's go. And we're basically adding layers of detail here. Okay. So, in your genome, you have not just, one, but a whole bunch of different heavy chain and light chain genes. And so you'll have different types of heavy chain. And so we'll call those like mu chains, or if it's IgM, or gamma chains if it's IgG. But you'll also have versions that have different specificities. And each of these is only the starting specificity of the antibody. It's just like a general type. And so if we're looking at the whole antibody here, we're just looking at the heavy chain region, there are going to be 3 regions, which are these red regions, where there's a lot of variability. Like some of these, you actually have 100% variability. So there are no 2 that are actually the same. That's just pretty cool. It looks as though it goes up to 110%, which always give 110%. But I think their little chart is just off here because that doesn't really make sense. And in terms of variability in the light chain, there's a little less variability, but it's also mostly clustered at those 3 positions. So there's 3 loops called hypervariable regions in the light chain, and 3 hypervariable regions, which are also loops in the heavy chain. And together, these are the bits that change, that make for different antibodies. And so what you're going to do, or what your immune system is going to do, because you don't have any control over this, is it is going to combine a light chain and a heavy chain at random, and it's going to test it out, see if it works. And it's going to combine all the other parts that make up these chains. If it makes an antibody that actually binds to something useful, then it will get what's called a second signal, which is like a kind of a return signal from other parts of the immune system that'll say, yes, you bound to the thing that I'm holding out that I recognize as dangerous. Good job. Yeah. And that second signal will cause the cell to divide. And so I'm being vague about what cell this is because this works for multiple cell types. This is the way T cells mature, and this is the way B cells mature. And there are lots of different kinds of T cells and lots of different steps in the maturation process of B cells. So yeah, that's the only reason I'm being a little bit cagey there, is that it applies to a lot of stuff. So it's a good general mechanism to get hold of. All right, and if you were to look, these are our same diagrams from the other half of the chapter. So here are the regions that are high variability. Here you can see on a sort of a ribbon diagram of the protein. So all this yellow bit is the protein. And you can see every protein is just like a big long spaghetti string that's folded up into some shape. And depending on which amino acids it has in what place, it will either, link up in a variety of ways. The 2 common ways are beta strand and alpha helix. And so here's a big long beta sheet, basically, that you can see here. And the little black square with a line in between, that's showing the disulfide bond that's going to bring this together. So the disulfide bond basically folds this sheet in half, so that you end up with a top half, which is like a beta sheet, and a bottom half, which is a beta sheet as well. And all 3 of the loops are going to end up on the hypervariable loops are going to end up on the same side. And so they're all going to be sort of brought together when you fold your sandwich in half. Yeah. Yep. And just like rolling up a taco or something. And you're going to end up with all that stuff right together at the end. And so it gives you a binding surface. Sometimes it's going to bind. Most of the time, it's not going to bind to whatever particular thing your body's dealing with at the moment. And that's okay. That cell tried its best, and now it needs to die, and another cell can come in and try its best. And that's how the immune system works. It's just a nonstop conveyor belt of, cells trying things out, doing their best to help you out, but kind of without any guidance, really. The only guidance is if they manage to just luck into a solution that happens to work and bind to whatever thing is being presented to them, then they get a signal which allows them to divide and then make further changes. And so they'll play around a little more with some of these variable loops. Okay, fine. So yeah, monoclonal antibodies just mean a specific version of an antibody that you can make a whole bunch of, exactly that same antibody. And as long as you have the genes, the particular version of each part of the antibody that have been put together in the right way, yeah, there's no limit to how much antibody you can make. It's just farming at that point. Yeah. And yeah, if it's a useful antibody, that could be very lucrative, or it could help a lot of people. And if it's not a very useful antibody, then it's okay. Yeah, it's easy to get a lot of different antibodies. That's very doable. Okay. Antibody-antigen interaction. So antigen is just something that is going to be recognized by, for example, a T cell receptor or an antibody or something like the CD1 protein, whatever. Just anything that the immune system can recognize in some way. Let's call it an antigen. And that's not a great definition, but it's kind of such a broad term that it's pretty hard to define any better than that. Think of the antigen as the bad guy in our little situation, and think of your immune system as the good guy, if you like. So there you go. There are different ways that your antibodies or T cell receptors or whatever, because they're all kind of built the same. They're genetically related to each other. Evolutionarily, we just kept copying the same things and slightly varying them. But you can have some little antigens that are very small that'll bind right down in the cleft there, which is great. And you can have some that'll sort of be able to stretch out in between the heavy chain and the light chain, like so. You'll have some that'll cover much wider areas like this, just sort of the entire top surface. And some of them where the antibody actually has some additions to it, which is a thing that can happen. There are some antibodies that'll have really long loops there, and they can fit like a little key into a slot. And so here you can see the antibody poking up and making contact, yeah, with a, I don't know, like a donut of antigen. It's got a hole in it, yeah. Or maybe it's like a bowling ball, I guess. The hole doesn't go all the way through, yeah. One finger bowling ball. I don't know. Metaphors only get you so far, right? Okay. How can antibodies interact and T cell receptors interact? Same way that proteins always interact. And here again, yeah, if you haven't had a ton of protein structure, this may seem like new and magical stuff. And if you have, this will just seem like, yeah, I already know this. So you can have electrostatic interactions where something positive and something negative will try to be close together. Yeah, which is fine. And that's just opposites attracting. You can have hydrogen bonds where you have sort of partially negative atoms. So like greedy things like oxygen, they're going to sort of pull the electrons around to one side. Oxygen doesn't share maybe quite as well as it should. And you have things like hydrogen that are not good at pulling electrons toward themselves. They tend to be a little bit positive. And so the sort of negative oxygen is going to interact with the sort of positive hydrogen. And you're actually going to develop a pretty strong little bond there. If I remember right, each hydrogen bond is they vary, but they're in the neighborhood of, -1 kcal per mole, if you're into amounts. Yeah, that's not something I'm going to test you on. But if you're just interested in figuring out roughly, what's an interaction look like in terms of energetics, or if you dabble in physics, yeah, go wild. Van der Waals forces, that's fine. These are things that can happen. They only operate at very, very short distances. So Van der Waals forces could help lock an antibody on. But if you're talking about guiding an antibody in at a distance, it's going to be electrostatics are most likely to do that, and also hydrophobic interactions can do that. So these are going to be parts of the protein that basically repel water. It's more complicated than that, but I think that's an okay crutch for us to use. And so you get two of these parts; they will stick to each other just to be away from the water. It's you may know people like this that just come together to shut out the rest of the world that doesn't understand them. I don't know. Yeah. And then the other thing, you can have our cation pi interactions. So you can have these electrons that have kind of a bigger range of territory that they can occupy. So they can kind of be anywhere around this particular ring. And those can interact with a positive ion, and you can have a great, strong interaction. There are a couple of amino acids, arginine and lysine, that would have a legitimate positive charge on a nitrogen in both of those. And so that would be your cation. And then in terms of pi bonding, you have a couple of amino acids, tyrosine and phenylalanine are the two that are easiest, but that would have electrons that are in one of those aromatic rings, and could make a cation pi interaction, just like it's shown here. And whatever, there are a bunch of marginal cases too, but this is the main stuff, and we don't need to worry about the marginal stuff right now. Let's stick with the thing that works most of the time, I would say. Okay, just showing how some of these interactions could take place. You can have a glutamine, the cutamine, and so this is going to have, it's going to be polar, but not really positive or negative. And so some of the polar residues are going to be a little bit positive or a little bit negative, and they're going to interact and actually hydrogen bond with residues that are a little bit the opposite. Yeah, because that's what a hydrogen bond is. So if we've got 3 of these bonds forming here, we've just made a basically a -3 kcal per mole interaction, which is decent. But usually when you see actual proteins coming together, usually it's going to be more like in the 10, -10 region. That would be more like a normal interaction. Because the cell's complicated and the body's complicated. There's a lot of stuff floating around in the blood, and you've got to be bound pretty tightly, or else the interaction will just happen and then fall off. It's just like 2 pool balls bouncing off each other. So, yeah, at least as I understand it. Yeah. Yeah, all right. Yes, let's keep going. So you can have antibodies, or in this case, they've just chopped an antibody in half, so they just have Fabs, antigen binding fragments. Good for them. And you can actually use this as a way to find out where the antigen or the antibody binds. So in this case, they've got a virus, which is our little ball at the center, and they are, so this is West Nile virus, which you remember maybe from back in the day. You would get sort of flocks of birds that would just drop off wires or just be dead all of a sudden, on the street for no good reason. And a lot of the time it was West Nile. It also was transmitted to humans and does various bad things to people. It can give you a pretty bad fever and do some other nasty stuff. Anyway, they got some antibodies that they know bind somewhere on there. They put them on here, and what do you know? The surface of this virus is actually made of repeating units. So it's the same proteins over and over again. They're kind of tiled over the surface like the tiles on your bathroom wall, probably. And so everywhere that there's the right protein, you can bind an FAB. And so there you go. You can get great little structures. The reason they don't tend to use full-on antibodies for studies like these is that antibodies have these flexible hinge regions. So the FAB may interact with the antigen, just so. Let's say both of the FAB parts interacted. But the tail end of it is going to be flopping around. It can be in any sort of shape. And the way they make these pictures is by finding a bunch of things that are exactly alike and then averaging them together to get a really high resolution picture of it. And if you have giant floppy FC regions, then you can't really get a good picture, and it just messes up your whole thing. So that's why they do this. That's why immunologists and people in general like FABs. Yeah, even though they're not like a real thing that actually exists and has a use inside the body, separate from an antibody anyway. All right, and then just mentioning in passing, because it's good to know, there are, so human antibodies all are multi-chain, 2-chain antibodies. And so they'll have the same light chain twice, and they'll have the same heavy chain twice. You can make in a laboratory versions of antibodies with, 2 different light chains, 2 different heavy chains, and all kinds of weird stuff. Journals are full of these things. But if we're talking about what your immune system can make all by itself, yeah, the answer is 2 of the same heavy chain, 2 of the same light chain. And that's the minimum antibody. And that would be like an IgG or an IgD or an IgE. IgA is 2 of those, back to back like that, linked together, and an IgM, the common form that you would see circulating in the bloodstream, is going to be 5 of those together, like a little star. Yep. There also exists in the world, just so you know about them, single chain antibodies. And so single chain antibodies are just going to be a heavy chain with no light chain. And this, we think, is the more primitive version. So we think that this was an evolutionary step up from animals that did not have something like antibodies. And then the next step up that made like a more sophisticated immune system. And the up here is totally arbitrary, but up means more like us, I think, generally. Higher, yeah, taxa are more related to us in general. That's just human bias, though. Let's see. But that was the next step, is basically just copying the top of a heavy chain, calling it a light chain, and preloading a whole bunch of copies of that thing in our genome. And so this is what you find in mammals for the most part, but in some mammals like these llamas and camels and alpacas, you get these single chain ones. And you get them in, dare I say, more primitive animals like sharks. Yeah. And sharks have been around since, I think the Ordovician, so almost 500 million years ago. Yeah, so sharks have been around. Yeah, they're about as primitive as you come. And things like this probably have not been around for more than about 10, 20 million years. Yeah, there you go. All right. That's section 2 out of the way. We only have one more section to go. This is so easy, right? Yeah, of course it is. It's not bad. And this is a supplement. Absolutely. Look at the book. Make sure you understand that. This is just giving you a second way to hopefully get some of that stuff. So, I don't know. Smash like and subscribe or something. Something. Yeah. You don't actually have to do that. These are not, if you're seeing ads on these are not ads that I am putting on these. This is just YouTube cashing in. So, yeah. All right. Stay classy, YouTube.